Okay, good evening, everyone. It's a pleasure to be here tonight. And I would like to thank Dr. Mirvat Esim and Dr. Nedli and all the organizing committee for inviting me today. The title of this talk is AI in Immunology, Lessons from Novel Therapeutics. This is an exciting time for the field of immunology. The COVID pandemic has focused the world's attention on the field, and some of the jargon has become familiar to the public. So just a little bit of background. The immune system maintains a healthy body through a complex network of cells, tissues, and organs. An important concept in the adaptive immune system is the specific recognition of foreign pathogens, such as bacteria and viruses, by the immune cells, which is a critical step for downstream mechanisms that eventually result in their clearance. How this immune recognition happens is similar to how you recognize familiar faces. Let's say you run into your friend Adam in the hallway. What makes you recognize that he's Adam? Maybe his characteristic curly hair or his brown eyes, but not so much his hand or feet. Similar to the eyes and hair, the characteristic parts on a foreign antigen or a foreign protein are called the immunogenic epitopes. The recognition of these specific epitopes by immune cells is an important step that triggers downstream effector mechanisms to fight off an infection or cancer. Immune cells such as B cells and T cells that belong to the specialized arm of the immune system, the adaptive immunity, are responsible for recognizing these immunogenic epitopes and eliciting an immune response. For the humoral immunity or B cell immunity, each epitope on the antigen is recognized by a specific B cell clone. For cell-mediated immunity or T cell immunity, which I'm more interested in this talk, antigens are chopped up into small fragments or peptides, and the immunogenic peptides or immunogenic epitopes are presented to T cells. This antigen or epitope recognition results in activation of either a B cell or T cell to fight off the infection. Understanding which epitopes are recognized by the immune system as immunogenic is important for understanding how the immune system works and which mechanisms are protective. It is important for developing effective interventions such as vaccines for infectious diseases, vaccines for cancer, cancer immunotherapy, specific therapeutics for autoimmune disorders, and for immunosilencing proteins of foreign origin for their use in humans. So an important question that scientists are trying to address is, what is the immune system seeing? What are the immunogenic epitopes that, when recognized by the immune system, can result in effective clearance of the infection or killing of cancer cells? So I will use vaccines as an example here, since they're one of the oldest and most well-known immunological public health interventions. Historically, prior knowledge of the important immunogenic epitopes was not necessary for the development of vaccines, at least early on. Variolation is a famous example. It was the practice of scratching the components of pustules from an individual who was infected with smallpox into the arm of a naive individual, and this would result in subsequent protection of the variolated individual against the disease. However, figuring out the protective immune mechanisms and the important immunogenic epitopes became necessary when the development of vaccines against certain infections was not so straightforward, such as for more complex pathogens as HIV, Hep C, and others, and complex diseases such as cancer. So the strategy for scientists was testing as many potential antigens and epitopes as possible, which can be done by mass spec or using overlapping peptides covering the whole protein for T-cell assays. Now, of course, these experiments are time-consuming, labor-intensive, and expensive, and this is where AI can provide the necessary tools for prediction of immunogenic epitopes and reducing the time and cost for epitope discovery. As mentioned earlier, an important step for recognition of antigens by T cells is their processing or fragmentation into peptides, and then presenting an immunogenic peptide in complex with an MHC molecule. This is what T cells are able to recognize, an immunogenic peptide on an MHC molecule. For the sake of this presentation, I will focus on MHC class I and cytotoxic T cells. The question is, which peptides get presented to T cells? What are the patterns of antigen processing? Where does the protein get cleaved? Which peptides bind with a higher affinity to MHC molecules, and which ones bind to each different type of MHC molecule? And if we know these processing patterns and binding affinities, do those factors dictate immunogenicity, and are these peptides immunogenic? Many different machine learning algorithms were developed to predict which epitopes on a given protein are likely to be T cell epitopes. Some of these algorithms predict MHC binding, so they predict the IC50 values for peptides binding to specific MHC molecules. But it's important to know that binding to MHC is necessary but not sufficient for recognition by T cells. Other prediction algorithms predict processing, which means they predict epitope candidates based upon the processing of peptides in the cell. And a third type of prediction algorithm would predict immunogenicity, which means they predict the relative ability of a peptide MHC complex to elicit an immune response. Most algorithms focus on binding affinities and antigen processing. Our group at Dr. Karen Anderson's lab at the Biodesign Center for Personalized Diagnostics at Arizona State University, where I did my PhD, developed a machine learning algorithm that predicts T-cell epitope immunogenicity. I would like to acknowledge the people that I had the privilege of working with on this project, Dr. Chowell and Dr. Krishna, who developed the algorithm, and Dr. Ferdowsi, who I worked with to use the algorithm for developing a safer gene editing tool. In the next few slides, I will walk you through how this algorithm was developed, what it does, and how we use it to develop a safer tool for gene therapy. To understand the biochemical basis of differences between peptides that are recognized by cytotoxic T cells as immunogenic epitopes and peptides that are non-immunogenic, over 34,000 peptides were retrieved from the IEDB, which is the Immune Epitope Database, a publicly available database hosting epitope prediction algorithms. Of these, 5,035 immunogenic epitopes that were 8 mers to 11 mers, they were non-redundant peptides. They were reported to be MHC1 binders and are referred to here as immunogenic. And 4,853 non-redundant peptides were non-MHC1 binders and are referred to here as non-immunogenic. The differences in amino acid composition and biochemical properties of these two groups were then studied. This includes differences in hydrophobicity, polarity, and side chain bulkiness. Before I show you the results, I would like to talk about the amino acid residues on immunogenic epitopes that are recognized to be important for binding. TCR contact residues are the residues that make contact with the TCR or the T cell receptor, and those are residues number 4, 7, and 8 for HLA-A2 restricted 9 mers. Anchor residues are important for binding to the MHC groove, and these are residues number 2 and 9 for HLA-A2 restricted 9 mers. So here, these are the amino acid residue positions on the peptides, and this is their mean hydrophobicity. Black squares are immunogenic peptides, and white squares are non-immunogenic peptides. What was found was that immunogenic 9-mer epitopes were significantly more hydrophobic than non-immunogenic 9-mer peptides at each amino acid residue. For HLA-A2 9-mers, if we compare hydrophobicity of anchor residues, which are number 2 and number 9, there is no significant difference in hydrophobicity. But if we compare the hydrophobicity of TCR contact residues number 4, 7, and 8, there is a significant difference between immunogenic and non-immunogenic peptides. This means that TCR contact residue hydrophobicity predicts peptide immunogenicity for MHC class I HLA-A2 restricted 9-mer epitopes. Now, TCR contact residue hydrophobicity was used to develop a prediction model for immunogenic T-cell epitopes, and the model was used to rank peptides that had been previously shown experimentally to be immunogenic. So this is the predicted rank of the peptides of a CMV or cytomegalovirus protein called PP65, and here for a dengue protein called dengue polyprotein. So this is the predicted rank according to an algorithm, and this is the immunogenic epitopes according to experimental data. The solid line is the TCR contact residue hydrophobicity algorithm that was developed by our group, and the dashed line is the algorithm that is hosted on the IEDB server. So as the line moves this way, this means predicting a higher rank for epitopes that are actually immunogenic. So this means that this machine learning algorithm, ANN Hydro, that was developed by our team does a better job at predicting immunogenicity of T-cell epitopes than the widely used IEDB algorithm, and this was published in PNAS. So then we use this T-cell epitope prediction algorithm to create an immunosilenced CRISPR-Cas9, as I will show in the next few slides. CRISPR is a bacterial defense mechanism against invading viruses. The 2020 Nobel Prize in Chemistry was awarded to Jennifer Doudna and Emmanuel Charpentier for using this system to develop a genome editing tool. That is a tool to rewrite the code of life. This system uses a nuclease called Cas9 that works like genetic scissors to cut specific DNA sequences, and then a modified sequence is introduced. The world interest in the CRISPR system can be demonstrated by the number of publications that have increased exponentially in the few years leading up to the 2020 Nobel Prize. We should remember, though, that there was all of this excitement about gene therapy in the 1990s. There were many success stories of treating life-threatening genetic disorders. However, the whole field backed off when 18-year-old Jesse Gelsinger died of a massive immune response to the adeno-associated viral vector component of the gene therapy that he was administered to treat a mild form of an X-linked genetic disorder. Therefore, an immune response against components of the gene therapy could have potential negative consequences. Many bacterial species possess a CRISPR-Cas9 system, but the most widely studied system is the one from Streptococcus pyogenes, or Strep pyogenes, a bacterial species that is part of the normal bacterial flora in humans, and it causes 700 million infections each year. Given the foreign nature of the Cas9 protein and the ubiquity of the bacteria that it is derived from, we hypothesized that healthy individuals have pre-existing T-cell immunity to Cas9. To investigate this, we predicted HLA-A2 restricted T-cell epitopes derived from SP-Cas9 or Strep pyogenes Cas9 using our ANN Hydro algorithm. There were two scores for each peptide: normalized binding score, or SB, which reflects its binding affinity to MHC, and the immunogenicity score, or SI, which reflects the hydrophobicity of TCR contact residues. So the best candidates would be expected to be in this quadrant here. They would have both high MHC binding, which is a low SB score, and more hydrophobicity, which is a high SI score. Now, these ones that were designated alpha, beta, gamma, and delta were the ones that we showed experimentally to be immunodominant and subdominant, as I will show in the next few slides. So here are the top 38 peptides of SP-Cas9 as predicted by our model and ANN Hydro, and these 4 peptides were in the top 5 of all predicted epitopes. Now, if we compare this to the commonly used publicly available IEDB model, IEDB ranked the 2 immunodominant epitopes alpha and beta as 5th and 14th, and the subdominant epitopes as 4th and 18th, while our model put all 4 peptides in the top 5 epitopes. We then synthesized the top 38 predicted peptides and used them to stimulate healthy donor T cells for 12 donors, and detected the response by an ELISpot, which is an assay that measures T-cell immune reactivity. So this heat map here shows the results of these ELISpot experiments. This is a map of the Cas9 protein. A darker color is higher reactivity of this peptide at this position on the protein for this donor. We found that the immunodominant epitopes were alpha and beta, and the subdominant epitopes were gamma and delta, and almost everyone reacted to beta. So now that we identified the immunodominant T-cell epitopes of SP-Cas9, we wanted to create a less immunogenic version of the protein. We hypothesized that targeted mutation of the anchor amino acid residues of the identified immunogenic epitopes would abolish specific T-cell immune responses. What if we cover Adam's eyes or change his hairstyle? Would he still be recognizable? If we make changes to the immunogenic epitopes on the SP-Cas9 protein, would it still be recognizable by the immune system? We synthesized these different versions of peptides alpha and beta through mutation of the second residue, 9th residue, or both, and we call them alpha 2, beta 2, alpha 9, beta 9, alpha 2 9, and beta 2 9. Then we use them to stimulate T cells. This is an ELISpot assay, and a higher number of spots in the well means higher T cell reactivity. The T cell response was abolished with the mutated versions, with 25-fold reduction from alpha to alpha 2 9 and 30-fold reduction from beta to beta 2 9, although mutation of only one of the two anchor residues was enough. Using pentamers that are specific to the T-cell clones that recognize epitope beta, the population of cells that were CD8 positive, pentamer positive, for beta was reduced from 3.09% to 0.3% when the mutated peptide was used to stimulate antigen-presenting cells. These experiments were done using peptides. We wanted to see the reduction in the immune response to the full Cas9 antigen. So antigen-presenting cells were transfected with mRNA encoding either wild-type Cas9, Cas9 alpha 2, Cas9 beta 2, or Cas9 alpha 2 beta 2. And these antigen-presenting cells were used to stimulate healthy donor T cells that are reactive to SP-Cas9. So these are the results of the ELISpot experiment. There was a significant reduction in the ELISpot reactivity when one anchor residue of epitope alpha was mutated, and a higher reduction with epitope beta, which was similar to the reduction seen when the second amino acid residues of both epitopes, alpha and beta, were mutated. So this shows that mutating a single amino acid residue of the Cas9 protein is sufficient to significantly reduce T-cell recognition. To examine the nuclease activity of Cas9 beta 2 and compare with wild-type Cas9, we targeted Cas9 beta 2 or wild-type Cas9 to an endogenous locus, EMX1, and measured percent indel formation or insertions deletions. So the data demonstrate that Cas9 beta 2 retains its nuclease capacity compared to wild-type Cas9. So next we determined whether Cas9 beta 2 can successfully recognize and bind its target DNA, leading to transcriptional modulation within the chromosomal contexts of endogenous genes. We transfected the cells with plasmids encoding either Cas9 beta 2 or wild-type Cas9 and 14 nucleotide guide RNAs against two different endogenous genes, TTN and MEOT. QRT-PCR analysis showed that Cas9 beta 2 successfully led to target gene expression. To further characterize Cas9 beta 2 specificity, we performed genome-wide RNA sequencing after targeting Cas9 beta 2 or wild-type Cas9 to the MEOT locus for transcriptional activation. And the results demonstrated no significant increase in undesired off-target activity by Cas9 beta 2 as compared to wild-type Cas9. And these results were published in Nature Communications and patented. So to summarize, machine learning algorithms...
5 minutes.
Okay.
5 minutes, Dr. Rod.
Thank you. Machine learning algorithms that predict immunogenic epitopes can provide the tools for understanding the immune system. Our model ANN Hydro does a better job at predicting T-cell epitope immunogenicity, and we use it to engineer CRISPR-Cas9 that is less immunogenic but is able to retain its function and specificity. Thank you for your attention.